Hello, this is Gina Piscatelli with a lecture on cell structure and organelle functions for week 3 of Anatomy and Physiology 1 at Madison Area Technical College. The reading is Chapter 3, Sections 1, 2, 7, 8, and 9, and you'll have a study guide question to help you take notes. That's study question number 13. The topic of this lecture really focuses on the structure of cells in a general way and functions of organelles located within cells. Cells have an intricate internal structure. This enables them to perform various functions in the body. And you can see this intricate structure in the figure shown on the slide. The green labels actin filaments. These are proteins that are part of an internal skeleton called the cytoskeleton. The blue labels the nucleus, and the red labels mitochondria, and this is where cellular energy is produced in the form of ATP. Now, cells do vary in shape, size, and internal structure, depending on which specific cell type you're talking about in the body and what function it performs. So for example, I've got 3 diagrams of specific cell types shown on this slide, and notice that the one on the left, the cells have elongated extensions. These are for conducting electrical signals. So this type of a cell is called a neuron. The picture in the middle contains epithelial cells. Notice that they're tightly adhered to one another, forming a nice protective sheet. So you find those lining organs and cavities. The third picture, the one on the right, is a picture of red blood cells. And notice that they're spherical and concave in the middle. And it's thought that that shape aids in their function of carrying oxygen. So cells do have a variety of shapes and a variety of internal structures due to their functions, but in this lecture we're going to focus on what they have in common. So even though there are different types of cells based on shape and function and maybe internal structure, some things that they all have in common include a nucleus that houses DNA, cytoplasm that consists of a viscous fluid called cytosol, small internal structures called organelles, and intracellular proteins that form a framework inside the cell. These are collectively called the cytoskeleton. And in addition, the boundary of the cell is made up of a phospholipid membrane, and it interacts with the environment in which the cell exists. Now there's one exception to this basic structure scheme, and that is that red blood cells do not have, well, I guess there's 2 exceptions. Red blood cells do not have a nucleus and DNA. The nucleus is lost when they're mature. And skeletal muscle cells have more than one nucleus, not just one. So there's a little bit of an exception, but these are the basic components of a cell. So we're going to start with the nucleus, and as I mentioned, most cells have just one. The cells that are anucleate are red blood cells. That means without a nucleus. Cells that are multinucleated are muscle cells. Not all muscle cells, but some muscle cells. So the structure of a nucleus includes the nuclear envelope, also called the nuclear membrane. It controls the passage of material into or out of the nucleus, and it protects the thread-like DNA, and the DNA is labeled chromatin in this picture, and I'll talk about what that means in a minute. Within the nucleus is a structure, it's a densely stained region, or darkly stained region, It's called the nucleolus. And this stains darkly due to the presence of RNA and protein, because this is where ribosomal RNA is made. Eventually, this ribosomal RNA will leave the nucleus and become part of a ribosome in the cytoplasm. And then, of course, there are nuclear pores that enable materials such as the ribosomal RNA to get in or out of the nucleus, but they're very tightly regulated what can get in and what can get out. And then finally, the thin dark lines are chromatin, and it's drawn this way because it's loosely coiled DNA, and it kind of surrounds the nucleolus. This slide shows a picture of the nuclear membrane or nuclear envelope in a lot more detail. Note that there are two phospholipid bilayers. So the nucleus has like two membranes. In contrast, the cell membrane is just one phospholipid bilayer. So I want to stress this by looking at just one of these. One second. Sorry about that. So I just want to stress one of these phospholipid bilayers on the next slide so that you understand that a bilayer, a phospholipid bilayer, has two phospholipids facing each other. If you recall in the lecture on lipids, we looked at the structure of phospholipids. They have a phosphate group attached to one fatty acid, which makes it polar. So that's what the blue balls are, the polar heads. And they also have two fatty acid chains that form the tails. They're shown as yellow here. And the reason these are labeled as nonpolar is because they consist mainly of carbon-hydrogen bonds. And these carbon-hydrogen bonds are nonpolar. So since the tails are nonpolar, they're hydrophobic. And therefore, they face each other to avoid the water-based cytoplasm and the nucleoplasm, or the fluid in the nucleus. We will discuss phospholipid structure again in the next lecture on the cell membrane. Within the nucleus are the 46 chromosomes of the human genome. These have been extracted from a dividing cell, shown on this slide here, and lined up into 23 pairs. Each member of a pair carries the same type of genetic information, such as eye color, hair color, or maybe coding for an enzyme. When cells divide, the chromosomes become tightly coiled. We call that supercoiled, so that they're visible under the microscope, and then you can see them as you see them here. Displaying chromosomes in this linear arrangement is known as a karyotype, and it helps geneticists look for damage and any abnormalities. So let's expand on the structure of chromosomes a little bit. So the genetic material in human cells always consists of 46 strands of DNA, and this DNA is wound around histone proteins labeled in the diagram. They look like little rocks. Commonly, we just refer to these 46 strands as chromosomes. We just call them 46 chromosomes. But in a non-dividing cell, a cell that is not dividing, the DNA within the chromosomes is loosely coiled around these histone proteins. And the chromosomes appear thread-like, and they're not supercoiled, so they're not visible under the microscope. This form of DNA in non-dividing cells is called chromatin. But in a dividing cell, the DNA and histone proteins supercoil, and become very tightly wound, and the chromosomes become compacted. The purpose of this is to prevent their fragmentation, because when a cell is dividing, the chromosomes get copied first, and then the copies have to be moved to two daughter cells. So fragmentation won't happen, or at least is least likely to happen. Okay, now let's look beyond the nucleus to the cytoplasm. The cytoplasm consists of all the material located between the nucleus and the cell membrane. This includes the viscous fluid called cytosol and many small structures called organelles. Now, the cytosol is made up mainly of water, 70 to 80% water, but it also contains many solutes like salts, proteins, lipids, carbohydrates, phosphate, enzymes, nucleic acids like RNA, ATP, lipid droplets, and of course organelles. So the organelles are suspended within the cytosol, and each one is specialized to perform a specific function in the cell. So we see in this picture a green organelle, an orange organelle, blue organelles, a white one, yellow, and we're going to talk about what those do. Some of the organelles are compartmentalized or enclosed by a phospholipid membrane, much like the nucleus is. Others are aggregates of compounds and molecules, just kind of clumped together. So let's start with the cytosol before we go to organelles. The functions of the cytosol, in addition to just being the fluid in which organelles are suspended, include a variety of cellular processes that will be performed by the proteins, enzymes, and solutes within the cytosol. So this is a site of metabolic reactions, both catabolic and anabolic reactions. So for example, the breakdown of glucose into pyruvate is a catabolic reaction that takes place in the cytosol, and we'll talk about the breakdown of glucose later. An anabolic reaction is the formation of ATP when ADP and phosphate are put together. So that also takes place in the cytosol. So now let's look at the different types of organelles contained within the cytoplasm. There are two basic structural types. One is those that are membrane bound or membranous, and the second is those that are not membrane bound and instead are just aggregates of proteins or other molecules without a phospholipid membrane. Those that have a membrane include the mitochondria, the rough and smooth endoplasmic reticulum, often called the rough ER and the smooth ER, the Golgi apparatus, which is also called the Golgi body or the Golgi complex, and then various vesicles, including lysosomes, peroxisomes, secretory vesicles, and transport vesicles. We'll look at everything but the peroxisomes. The organelles that don't have a membrane include ribosomes, which are aggregates of RNA and protein that I'll talk about later, centrioles, and the centrosome. The centrosome is where microtubules are made, and we'll talk about that in more detail later. Cytoskeletal proteins, and cilia and flagella. So all of those are examples of, I guess you could call them organelles, but they don't have membranes. So we'll start with mitochondria going down the list. And most types of cells have many mitochondria, not just one, and one is called a mitochondrion, O-N. These organelles contain enzymes that contribute to the production of ATP, otherwise known as adenosine triphosphate. Therefore, we often say the function of mitochondria is ATP synthesis. Now, the structure of a mitochondrion is unique. It has two phospholipid bilayer membranes. Well, that's a lot like the nucleus. One is called the outer membrane, the other is called the inner membrane, and the space between the two is called the intermembrane space. The innermost space, encapsulated by the inner membrane, is called the matrix. Not labeled on there. Notice that the intermembrane space contains ribosomes. Those are labeled, they're green little balls. And this enables the mitochondria to synthesize proteins. Now, the proteins are usually enzymes needed to make the ATP, which is the main function of the mitochondrion. So therefore, mitochondria can do protein synthesis. Also notice that the matrix contains a single circular chromosome. Well, it shows many here, but it's exactly the same information, only a limited number of genes. And this chromosome, although it's been copied in this picture, contains genes that encode for those enzymes that the ribosomes need to make. And so those enzymes, again, are required for ATP synthesis. So for a mitochondrion to make ATP, it's going to need some enzymes. Some of the enzymes are coded by this mitochondrial DNA, but there are also nuclear genes that code for some other enzymes that are also needed. So the function of mitochondria is dependent on both nuclear and mitochondrial DNA. Lastly, notice that the inner membrane has foldings or invaginations. These invaginations are called crista, and they contain one important enzyme. They contain a number of things, but one important enzyme I'll mention now is called ATP synthase. That is the enzyme that's needed for the final step of ATP synthesis. So when we talk about ATP synthesis, we'll talk about that inner mitochondrial membrane and the crista once again. Now on to the endoplasmic reticulum, or commonly called the ER. This word and our term endoplasmic reticulum comes from the fact that reticulum means network and endoplasmic means inside the cytoplasm. So its name, endoplasmic reticulum, comes from the fact that it's an extensive membranous network inside a cell's cytoplasm. The membranous system contains sacs and tubules, shown in blue. The spaces inside the sacs are called cisterns or cisterna. Sometimes I guess you could call it just a space or a cavity, but cisterna is the correct term. Now there are two divisions of the ER. One is the rough ER that has ribosomes attached to it, and it got its name because looking under the microscope it reminded somebody of rough sandpaper. And then the smooth ER, which doesn't have any ribosomes. Ribosomes are absent. The two divisions of the ER have very separate functions. The function of the rough ER is associated with proteins, and the function of the smooth ER is associated with lipids. So first I want to talk about the rough ER. So the rough ER helps with protein trafficking after protein synthesis because the attached ribosomes are synthesizing proteins, and as the proteins become longer, they're inserted into the rough ER cistern. Once the proteins enter the rough ER, they can be modified, which we call glycosylation, the carbohydrates added. And then they're trafficked throughout that membranous network and put into vesicles called transport vesicles. Ultimately, all the proteins that are made by ribosomes on the rough ER and put into the rough ER are destined to go to the cell's membrane, and we'll talk about that later. The proteins will either be secreted, that means released from the cell, or be attached to the cell's membrane. The function of the smooth ER I mentioned has to do with lipids. Another function of the smooth ER is detoxification and storing calcium. So the smooth ER contains enzymes needed to synthesize cholesterol, triglycerides, steroids, all those lipids that we already learned about. The lipids made inside the smooth ER will be put into transport vesicles as well. Separate ones, but not the same ones as the proteins, but they'll be put into transport vesicles. Additionally, the smooth ER is a site where calcium can be stored and released in vesicles or through pores. Another that happens in muscle cells. And another thing that the ER can do, the smooth ER can do, is alter certain drugs and pesticides. There are enzymes that help denature them, these drugs and pesticides, to make them less harmful. So this slide shows the process by which transport vesicles are formed from the endoplasmic reticulum. In this case, the picture shows the rough ER, but it could also happen on the smooth ER. So the ER membrane invaginates or constricts. A vesicle buds off of the ER, and any proteins or lipids that were in that part of the cistern will get trapped, and when a vesicle is formed, the vesicle will contain the protein because we're in the rough ER. So if it's a smooth ER, of course, the vesicle will have lipids inside. Now eventually this transport vesicle will migrate in the cell. It can move along cytoskeletal proteins, or it can just sort of diffuse. And depending on what's inside of it, it might go to the Golgi apparatus, and I'll tell you what happens when it gets there in a minute, or it could go directly to the cell's membrane, or it could go to a lysosome. So let's look at the Golgi so we see the process of this migrating transport vesicle going to the Golgi apparatus. So first, the structure of the Golgi apparatus. It's shown here in green, and it's a series of hollow flattened sacs stacked on top of each other. Usually 3, but it varies. The side closest to the ER is called the cis face, and the side facing the outer cell membrane is called the trans face. The membranes of transport vesicles from the ER fuse with the membranes of the Golgi's cis face. So when a transport vesicle bumps up against that cis face membrane, all the phospholipids fuse together. When this happens, the proteins or the lipids in the vesicle move into the cistern of the Golgi, and they will then move throughout these adjacent sacs and be sorted and in some case modified again by glycosylation. Finally, the sorted mature protein or lipid or both will be put into a secretory vesicle, because a secretory vesicle will bud off of the trans face of the Golgi apparatus. Due to its role in sorting proteins and lipids, the Golgi is sometimes called the post office of the cell. And it sorts these proteins and lipids based on what they are, what kind they are, and by where they're ultimately destined to go. So I just want to review this process, and we'll look at the Golgi complex one more time. First, a transport vesicle from the ER merges with the cis face of the Golgi. I said fuse before, but fuse and merge, same thing. And the contents of the transport vesicle are dumped into the lumen of the first Golgi sac. These lipids or proteins are moving throughout the Golgi, and they're exposed to enzymes inside the Golgi. These enzymes can add carbohydrate. Not every single lipid and protein will have the carbohydrate added. It depends on their sequence of subunits. But the process is called glycosylation when carbohydrate is added. Vesicles bud off of the Golgi's cis sac, move a very short distance, and then fuse with the next Golgi sac. And then the same thing happens again. Another little tiny vesicle fuses off of the middle sac, moves a very short distance, and enters or fuses with the trans-facing sac. Regardless, those things aren't as important as the final step, which is at the trans face, a secretory vesicle buds off, and it will contain the proteins and lipids destined to go to a certain place. Now, these secretory vesicles can move within the cytoplasm to fuse with lysosomes or with the cell membrane. If a secretory vesicle fuses with the cell membrane, its contents will either be attached to the cell membrane or released into the extracellular fluid surrounding the cell. Now let's look at the role of ribosomes, since they were involved with the rough ER. Ribosomes are an example of non-membrane bound organelles. And there are two types: free ribosomes that are suspended in the cytosol, and bound ribosomes that are attached to the rough ER. Both types have the same exact structure. They're both made up of two subunits. In this picture, one subunit is dark purple and the other subunit is like lilac or pink. And each of these subunits, or both of the subunits, contain ribosomal RNA that's made in the nucleolus and protein. And that protein has been made by other ribosomes. But the protein and the RNA bunch up together and form these ribosomes. Now we'll look at the function. Now there's similarities and differences between free and bound ribosomes. The similarities first. Both free and bound ribosomes perform protein synthesis. That is, a ribosome can assemble amino acids to make a polypeptide. And it does this by reading a copy of a gene, shown in green on this picture. The copy was originally made in the nucleus, because the original gene is in the nucleus. And that gene codes for a particular protein. So now the ribosome is reading it, and it will assemble a polypeptide according to the instructions of that gene. And in the picture, the polypeptide that's being made looks like a pearl necklace, white circles. Now the differences between free and bound ribosomes. The free ribosomes always remain suspended in the cytosol. They are not, I should say not ever, but they are not attached to the ER. So they make proteins that are going to stay inside the cell. They are inside the cytoplasm, and all the proteins they make stay inside the cell. Bound ribosomes are a little bit different. The bound ribosomes on the rough ER synthesize proteins that will go to the Golgi apparatus and then the cell membrane. This actually occurs, strangely enough, when a free ribosome docks onto the rough ER. So the next slide shows that process. So we have a purple ribosome in the cytoplasm reading a copy of a gene that's green. It's labeled messenger RNA, and we'll talk about that later. So it always begins, this protein synthesis always begins with a free ribosome. But if there's a certain sequence in the polypeptide, that causes the free ribosome to dock onto the ER, making it rough ER and turning the ribosome, of course, into a bound ribosome. Now, as the ribosome continues to read the gene, the polypeptide that's synthesized is inserted into the ER, as we mentioned before. Eventually, as we know, this purple protein that's been made in the rough ER will be in a transport vesicle when the transport vesicle buds off, and it will travel to the Golgi. So in summary, the rough ER and smooth ER have certain functions related to the functions of the ribosomes and the Golgi body. If we look at the diagram in the upper right corner, notice that the smooth ER without ribosomes is where lipids are made. The ribosomes on the rough ER synthesize proteins that are inserted into the lumen of the rough ER. Now, both lipids and proteins can leave the endoplasmic reticulum in transport vesicles that bud off. In the picture, we just see a protein, or we just see a vesicle budding off of the rough ER containing a protein. This transport vesicle then travels to the cis face of the Golgi body, fuses with the Golgi's membrane at the cis face, and releases its contents into the first sac of the Golgi. As that protein moves throughout the adjacent sacs of the Golgi body, it can be altered by some enzymes in the Golgi. Carbohydrate can be added, and that process is called glycosylation. At the transface of the Golgi body, secretory vesicles bud off, and these secretory vesicles contain the modified protein. The secretory vesicle then will travel to the cell membrane and fuse to the cell membrane. In some cases, the protein will just stay right on the cell membrane, and in other cases, the protein will be released into the extracellular fluid. So I haven't really mentioned lysosomes yet, but some secretory vesicles that bud off of the transface of the Golgi body actually contain worn out compounds that were parts of organelles that got old, and they need to actually be broken down. So the Golgi body can put enzymes into these secretory vesicles that are called lysozyme. That's what the enzymes are called. And they'll break down carbohydrates, proteins, lipids, and nucleic acids. So the Golgi can also produce what are called kind of degradative vesicles. Vesicles that fuse with something else and break it down into a usable form for the cell. This happens in another instance. For example, imagine a cell ingesting a large compound or maybe even another cell to get nutrients. Well, what it has ingested is not in usable form. but the lysosome can fuse with it, with this ingested structure, and break down the material so that it'll be usable for a cell. So we've covered a lot of cellular structure and function, including the nucleus, the nucleolus, the nuclear envelope and pores, and DNA. We've also talked about mitochondria, rough and smooth ER, the Golgi body, ribosomes, and lysosomes. It might be a good opportunity for you to pause this and answer your study guide question. Or you can continue. When you're ready, though, proceed with the rest of the presentation, which discusses the organelles that we have left that don't have membranes, including the centrosome and centrioles, the cytoskeleton, cilia, and flagella. So I'm going to start with the centrosome and the centrioles. What's noticeable inside of a cell are paired centrioles. They look a little bit like bits of licorice to me. They are paired, there's two of them, and they are made up of linear structures called microtubules. Now, where did these microtubules come from? They're made up of a protein called tubulin that has been assembled by this region that they exist in called the centrosome. So the region surrounding the centrioles is called the centrosome, or maybe more technically, it's called the microtubule synthesizing and organizing center. So microtubules are important not just to make up centrioles, which I haven't talked about function yet, but they're important to make up centrioles. But microtubules also compose parts of the cytoskeleton that give the cell shape and structure. They help during cell division by forming this thing called a spindle apparatus. And they can make up cilia or flagella depending on the cell type. And that has to do with movement. So let me show you the structure of centrioles first. So remember, every microtubule that we're going to see from now on has been organized and assembled by the centrosome. So centrioles are paired. There's always two, but this picture shows one. Okay, and they're made up of a protein called tubulin that aggregates to form these long tubes. The tubes are called microtubules, I guess, because they're small. And in centrioles, there are three microtubules kind of adhered together, and we call that a triplet. In centrioles, there are 9 triplets every time, always. And there's some other proteins that keep them all held together. Now, the reason centrioles are important is because they become the basis for which a big structure is formed in a cell that's dividing to help move chromosomes. And we'll talk about that when we do cell division. I didn't want to overwhelm you right now with that. Now, another part of the cell that has microtubules is the cytoskeleton. And notice that in this picture they're green, so the microtubules are green. They usually are single or double microtubules twisted together. And I'll talk about what they do in a second. But since they're part of the cytoskeleton, let's look at 3 components of the cytoskeleton, or the structure of the cytoskeleton. One is the microtubules, the green ones. The second are intermediate filaments, which are orange. And then the third and final are actin filaments, which are red. Now, all of these are elongated structures made up of protein. Microtubules, as you know, are made up of tubulin. Intermediate filaments are often made up of the protein keratin. And actin filaments are made up of the protein actin. All of these long tubes can become longer or shorter depending on what the cell needs. As you can imagine, when a cell grows, they're going to have to become longer. So notice that the microtubules aren't branched. They're suspended within kind of the center of the cell more, and they're the largest in diameter. Also notice that the intermediate filaments have an elaborate framework. This provides cell shape and scaffolding to give the cell support. And finally, notice that the actin filaments are adjacent to the cell membrane, and that's going to play a role in what they do. So let's look at the functions of these different cytoskeleton proteins. Microtubules help with the movement of materials inside of a cell. So this picture on the top shows a green microtubule made up of tubulin subunits. So it's a long green microtubule. And walking along it are some other motor proteins carrying a vesicle, like a transport vesicle or a secretory vesicle. So microtubules help with movement of materials inside of a cell. Intermediate filaments are just about strength, support, shape. But actin filaments are also involved with some kind of movement. But in this case, it's not movement of substances in the cytosol. Instead, it's movement of the entire cell. And it happens as the cell pinches or crawls, like expand, contract on one end. So in the case of muscle cells, actin is part of muscle contraction, but I'm thinking more along the lines of something called amoeboid movement. So this amoeboid movement happens when a white blood cell leaves your bloodstream to go to an infected or inflamed tissue somewhere in your body. So remember that actin filaments lie along the cell membrane. And if the actin filaments are short, the cell's pretty static, it's just sitting there. But then if more actin aggregates together, they elongate. And since they elongate, the cell membrane has to elongate with them. And that's what helps the cell extend in one direction. But of course, it's got to bring up the hind side, if you will, and it has to kind of pull the backside towards the leading edge. So actin filaments are involved with the movement of an entire cell. Okay, now we're going to go back to microtubules again. Again, microtubules are all made by the centrosome. And another place we find microtubules are in cilia and flagella. So in this case, these two cell structures, cilia and flagella, have 9 pairs of microtubules surrounding one central pair in the middle. So 9 outer doublets and 1 central doublet. And there's another protein involved with this, with their function. It's called dynein. The same is true for cilia and flagella. We have to keep that in mind. However, this dynein and other motor proteins might vary because they move differently. So first, let's talk about where you find cilia and flagella. In the human body, the only place we see a flagellum is on sperm cells. And cilia is found in a number of places, but the most commonly talked about is in your respiratory tract. It helps us move mucus along the surface of cells. So again, the flagellum and the cilia are made up of microtubules and another motor protein. But notice that in the picture for the flagellum, there's only one on the cell, on the sperm cell, and it kind of rotates. It like swivels, spins, if you will. But in a cilia, there are usually many cilia on the surface of a cell, and we call it undulate. but they move in a wave-like fashion, back and forth, left and right. And that propels fluid and any other substances along the surface of the cell. That's all for cell structure and function, and thank you for listening.